Welcome to the STEM Pathways podcast, where we delve into the fascinating world of science, technology, engineering, and math. I'm your host, Rohan Sharma, a passionate explorer of all things STEM. Each episode, we bring you captivating stories, cutting edge research, and inspiring interviews with experts and enthusiasts who are pushing the boundaries of innovation. Whether you're a student, a professional, or just curious about how the world works, join us as we unlock the mysteries of the universe and explore the endless possibilities of STEM. Let's get started. Today, I'll be interviewing Christopher Owens, who holds a Ph.D. in Microbiology and Molecular Genetics from Harvard. How are you doing today?
I'm doing great. How are you doing?
I'm also doing good. So getting started, I want to ask you what made you want to do an associate's degree?
Like many things in life, it was something of circumstance, I guess. So out of high school, I actually I had a free ride scholarship to New Mexico State. I grew up in New Mexico. And it was only a 2-year program at that satellite university. And I got to the end of my 2 years and I had no idea what I wanted to major in. I could have gone into geology. I could have gone into psychology, um, genetics, microbiology. I was interested in everything. And so I ended up getting an associate's simply just to lock in my programs that I'd taken. That was really the only reason I did that. And then from there, because I had no idea what I wanted to do, I took a year off. And I didn't feel like I was slowing down my career too much because I graduated high school a year early. So I was just normalizing. So during that year off, I spent my weekends at the library because the internet didn't exist back then, and researched careers in science, and tried to figure out what it was I wanted to major in. And at the end of that year, I applied to several different colleges and I transferred to Knox College. Knox College was in the middle of Illinois, and but they had a fantastic biochemistry program, and they gave me the most money, so that's where I ended up going. And that's how I ended up transitioning towards my bachelor's.
Oh, what was your experience like while majoring in biochemistry?
Well, it was interesting because after my first year, I had a sudden, oh wait, maybe I screwed up. Maybe I picked the wrong major. Maybe I really wanted psychology. And so then I scrambled to take a bunch of psychology courses, and I got to the end of those and decided, no, that's not real science. So I went back into biochemistry, and I almost added organic chemistry because I loved organic chemistry. That was a lot of fun too. But
I don't hear a lot of people saying they love organic chemistry.
oh, it's amazing. It's so incredible that you can predict what structures you will end up getting based on light, heat, chemicals added to a reaction, and you can actually literally predict the structure. It's great. It's super fun, but it is challenging. So,
Yeah. So what made you decide to go ahead and do a master's?
the master's. So when I was researching graduate programs, I came across Harvard. I was looking at Harvard because everybody looks at Harvard. I basically had a tiered system where I basically picked the top 5 universities, picked one or two of those, picked one or two of the next 5 down on the list, and then the next 5, and that's how I sort of selected my university. Um, but Harvard had this great program where first you'd have to get into Harvard, but then once you were there, you'd have to apply to this program where after your first year in grad school, they took you out of your PhD program and dropped you into Harvard Medical School as a medical student. And you spent the next year and 3/4 basically as a medical student. And um, and I ended up getting into Harvard. I was happy with that. went, applied to that program, got into that program. And at the end of that time in medical school, they gave me a master's in medical science, and then they put me back into my PhD program. But it was great. It was a great experience. I learned a lot about medicine and pathophysiology of diseases, and was able to, over the course of my career, I've been able to get up to speed very quickly on new disease areas, basically because I had that experience in medical school. So,
Can
yeah.
you talk a little more specifically about what your research was about?
Yeah. So around the time when I was looking into what I wanted to major in, there was a book that came out called The Hot Zone. They also made a movie about it, but it was all about Ebola virus, and I was super interested in Ebola virus. So it was just incredible to me that something so tiny with so few genes could cause just a horrifying death in humans, and so I really wanted to look at viruses. And so when I actually applied to Harvard, I applied to both the virology program as well as the BBS program. I ended up ultimately choosing the BBS program. It stands for Biological Biomedical Sciences. And just largely because it was a larger umbrella program and I could still work in a virology lab if I wanted. But given the fact that I hate making decisions, and and the possibility that I might be interested in something other than virology after spending a little time in a couple of courses, I chose the BBS program because it just gave me more flexibility. But so I ended up rotating through a couple different labs. One of them was studying leishmania and malaria in the School of Public Health. I thought that was interesting, but ultimately decided against it. Did a few other rotations, and then ended up in Joe Sadrowski's lab because he studied HIV, and HIV was very hot back then. All HIV research was just getting money dumped on it, and so it just seemed like there was an opportunity there to make a difference and to learn something about HIV biology, and that's the lab I chose. And there was an interesting phenomenon when I joined the lab with HIV, where you could infect human cells with HIV, but you couldn't infect monkey cells with HIV. And I mean, we're 98, 99% identical to chimps and a little less so to other species of monkeys. But I mean, it just seemed to me quite odd that you couldn't infect those cells, because you could take SIV, simian immunodeficiency virus, that infects monkeys, and you could still infect human cells with it. And so it just didn't make any sense that it was just a black box. Nobody understood what was going on. And I decided that sounded really interesting. In retrospect, it's a really stupid project for a graduate student to undertake, because black box projects are more suited to postdocs. Whereas I was basically saying my PhD, my ability to get a PhD is dependent on me solving this problem. And if I hadn't been able to solve that problem, I wouldn't have gotten a PhD. So it's not generally those types of projects. You want something that has a concrete answer and methods to answer questions. And mine was like a big question mark. And it just was, in retrospect, not the best choice, but we solved it. So we figured out, I ended up creating a bunch of lentiviral vectors that had pieces of SIV. And I was swapping in pieces of SIV into the genome of HIV and created these vectors, and would infect cells with them and see, do they infect human cells still? Do they infect monkey cells? No? Okay, swap in a new piece. And so did that until we actually identified a piece that could actually overcome this block. And so it seemed to me that the block probably was not caused by a protein in monkey cells that was missing. It was just absence. It just didn't seem to make sense that you could overcome the block if there was a protein it needed. always possible that it's true, but it just seemed to me that from some of my other experiments, it seemed to me that there was a protein in the monkey cell that was actively blocking HIV infection right after it entered the cell. Because if I created concentrated lots of HIV and just dumped it on monkey cells, it was a vector expressing GFP. I could actually overcome the block. And so, my thinking was that we were saturating something inside the cell, and there was something in there that was actively blocking HIV. And so, not only did we ultimately identify specific portions of the HIV capsid protein that you could mutate to SIV sequence, and then it was no longer blocked, we also identified the cellular protein that was actually blocking the virus. And that was it. I was done, tied the bow, and and got my PhD. But yeah, that was my research.
Oh, did that discovery lead to anything?
Yeah, it catalyzed a whole new field of HIV research. I mean, 1 year later, that's all anybody was working on. That TRIM5 alpha and various variants found in all the different monkey species, and there were people working on it before. It's just it wasn't everybody. It just seems like after that discovery, that was it. That's all anyone was talking about and working on. It was pretty cool.
Are people still working on that to this day?
I doubt it. I haven't kept up with the field. I mean, I graduated my PhD in 2004.
Yeah.
So it's been 20 years. I don't know if anyone's I don't know if anyone's still I'm sure they're still working on it, but it's probably not nearly as many as were, but
Were there any
yeah.
treatments related to that?
No, nothing came of it. I believe one of the reasons is that it turns out that humans cells also express the same protein, TRIM5 alpha, and it's got a different amino acid at a point that's important for recognizing capsid protein. And I think it's undruggable is basically is basically what it comes down to. Is it'd be very difficult to drug. Although that said, technology has gotten to the point where it could be possible to drug it, I would think, but nothing's come of it since.
Okay.
Yeah, but it would be cool to be able to turn human TRIM5 alpha into resembling um, chimpanzee TRIM5 alpha or rhesus macaque TRIM5 alpha or something like that to get it to block HIV.
Yeah.
Yeah.
So talking from my limited AP bio knowledge, um, so you're saying there's one amino acid in this protein that's different in humans compared to monkeys, right?
Well,
And
there's
that,
other differences, but just in that active site, turns out that
yeah,
I think it comes down to a single amino acid difference that's important. That's the reason why human TRIM5 doesn't block HIV and the rhesus macaque version does.
hmm. And then
So
are you saying like maybe with present day like gene editing and other tools, they could
yeah,
potentially
you
be
could
able
do
to change
that.
that?
Yeah, you could certainly you could certainly use gene editing to like you could do base editing or you could do prime editing and change that.
Yeah.
Yeah,
All right,
you
so
could.
moving on then.
Yeah.
Uh, what was your first job out of your PhD?
I went into pharmaceutical consulting. Um, I was a director of the Harvard Biotech Club at the time, and we would have career fairs and we'd have um, invited guest speakers who would talk about, what they did, whether they were in consulting or research for industry or, patent law, things like that, alternative careers for scientists with a PhD. And consulting sounded interesting to me. I was a little naive at the time because I did not realize that not all consulting companies really do the same work. And um, jumped into one company, Trinity Partners, and I thought I would be doing something much different than what I was doing. And in my first 2 weeks there, I realized I think I'd made a mistake. Um, and it's largely just because the focus of the firm, it's different now, but the focus of the firm then was on much later stage companies, helping them commercialize their products, helping them with marketing and with, gaining market share and things like that, which I didn't felt you need a PhD for.
Were you still
Um,
interested in like the more cutting edge research aspects?
yeah, yeah.
Yeah.
So, and throughout throughout my career, I have actually bounced back and forth between consulting and research. And it's because I have an interest in business, but I also have an interest in science. I would say I'm probably more 65% interested in science and 35% interested in business. So when it gets too businessy, I lose interest.
Yeah.
Um,
No
uh,
offense, but I talked to one person and then he's involved in consulting too.
uh-huh.
And then he was saying he doesn't like working with PhDs because they're too interested in the research aspect
Yeah,
and not
uh,
enough in the money making.
yeah, yeah.
But
And not even the money making. It's they're just less interested in business fundamentals.
yeah,
I'm not speaking for everybody, but for myself, I'm certainly less interested in like Porter's five forces and all of that kind of stuff. It's just not my thing. I'm much more interested in some new novel technology. But but yeah, so I went into that. I was there for a year and a half while I at Trinity Partners while I tried to figure out what I was going to do next. Um, and I really ultimately came down to deciding I probably needed to go back to research. And I almost did a postdoc, but ended up running into somebody at a networking event who worked for combinatorics, and he was telling me that they were thinking of getting into either Hep B or hepatitis C, and they needed someone to really consult them on the side as to what they would need to put in place in order to, run one of those programs. What were they going to need? What are the costs? What kind of reagents? They just really needed someone to sort of scope all of that out. And so they hired me on the side. I was just basically moonlighting on the weekends, just looking into into these two therapeutic areas, these two viral diseases. And um, about 3 months later, they offered me a job. And so I left consulting, I left Trinity, and went to combinatorics, built up my own team, was able to hire my own team, and was given a lot of free rein to run it myself. And we ended up pursuing Hep C first instead of Hep B. Um, and we had a lot of success with identifying some combination drugs. Combinatorics' whole principle was you could take already approved drugs that have been in humans. We know they're safe. We know they're effective for one indication, but they may target a certain pathway that interacts downstream with another pathway. And if you were to use another already approved drug to target that second pathway, they would synergize with each other and impact a whole new disease. And that was the idea behind the company. And so we were searching for combinations of drugs that would have an impact on hepatitis C replication. So
yeah. So what was your next job?
from there, I went to Enanta Pharmaceuticals, and I was there for 7.5 years. And it was because they were in Hep C, and it looked like combinatorics was getting out of Hep C. And I wanted to stay in the same indication. And so worked on developing a number of drugs there for Hep C, and some of them made it into humans, went on to clinical trials. And then we started looking at other indications like respiratory syncytial virus and parainfluenza virus and Hep B. I don't think we looked at HIV. Yeah, and I think that's pretty much it. And then I was there, I was there for about 7.5 years, and that was a good company to work for.
How different is doing research in a company versus at a university?
Got more money. It's quite different. And that's actually something that a lot of academics don't appreciate when they move to do research for industry. They like you'll see companies that are started by academics, and everyone there has this very academic mindset of, oh, this is a very interesting phenomenon. Let's pursue this. Oh, this is really cool, and they have all these little side projects. That's not what you do in industry. You have a goal. Your goal is to produce a therapeutic that has a meaningful impact on a patient's symptoms and disease. And that's your primary goal. And often you don't want to spend a lot of time on all the little side projects, because it might end up telling you something that negatively impacts your lead program and prevents it from going further because it raises concerns with the FDA. And you have to disclose everything that you discover as you're doing your research to the FDA. So generally it's frowned upon to pursue these side projects. But and there's just more money. You don't have to write grants unless you write SBIR grants, things like small business grants. But for the most part, you don't have to deal with that. And the equipment's nice, it's new, reagents are great. You don't have to make your own buffers, you just buy them. Like that's really the difference. But yeah, I liked working for industry.
Yeah. So what was your job after that?
So I had noticed that for several years that it appeared that there were very few viruses left to develop a drug for. There just weren't that many left that caused significant health issues in people and in populations of people that could afford a drug that you might actually develop for them. And so that kind of, yeah, dengue impacts a lot of people around the world, but it's generally in areas where people don't have a lot of money. And until it impacts the US population, which it will as global warming continues pushing dengue further and further north, it's just not something companies have spent a lot of money on, because they're not going to get a return on their investment. So there are companies that do, like Novartis has or they did, I don't know if they still have it, but their Institute for Tropical Diseases, where they do pursue research against things like that. Just trying to help the community, and it's a nice thing they do, because they already make profit on all their other drugs. But I'd noticed that there were just very few viral diseases left to target, and this is before COVID.
Yeah.
And so I wanted to get out of targeting viruses. I kept trying to find a way out of it. And that's one thing they never tell you, is that it can be very easy to get pigeonholed by industry. So once you've worked on something for a period of time, even though you have a PhD, and that PhD training enables you to conduct research in any area, for any indication, for any area of medical research. Once you've done something for long enough in a particular area, it's almost like they say, nope, that's all you know. You can't work on anything else. But high throughput screening to identify drugs for a virus is very similar to high throughput screening to identify drugs that can target cancer. And it just, it really makes no sense to me why scientists get so pigeonholed in industry. So that's always one thing to be careful of. Choose a broad enough area. Like I really probably should have chosen cancer. I mean, cancer, 70% of the companies, the pharmaceutical companies, biopharma companies out there are oncology companies. So if you want a job in industry, probably should go into oncology. So it's just something to keep in mind. But let's see. Wait, what was the question? I got sidetracked.
Or I would just ask you like what your next job was.
Oh, my next job. Sorry.
But, yeah.
So anyways, I was trying to get out of viruses. Ended up had a headhunter contact me and ask me about working in CRISPR, developing a CRISPR therapy. Of course, it was for hepatitis B, which is another virus. But my thinking was, well, this is a whole new modality. It was very early at the time. And I thought, this might be an opportunity to use my experience developing a CRISPR therapy to then transition into a different disease area. So I felt like that might be a way to do it. And so I accepted I applied for the job, got it, accepted it. And we were developing an AAV delivered CRISPR therapy for hepatitis B, or could have been LNP at the time. We were only working on that 6 months, and then that program ended up getting canceled because hepatitis B integrates randomly throughout a patient's genome during a chronic infection. And so the idea of taking a blunt end cutting restriction enzyme and targeting the same sequence spread across multiple chromosomes was probably a bad idea. It had a serious translocation risk. So we pivoted to herpes. And so there had been a pilot program ongoing before I joined to look at using CRISPR to target herpes. And we had an SBIR grant for that at Editas Medicine. And so we worked on that for a while. Herpes can cause blindness. So patients who get herpes, it can migrate back into their trigeminal ganglion, hide out, go latent, and then come back out an ocular route and infect their cornea. And after repeated rounds of infecting the cornea, it ends up just destroying the cornea and people just go blind. So that was really what we were pursuing, working on. And we did that for a while, about a year and a half. And then I ended up getting laid off, largely because the program wasn't of interest to our corporate partner, Allergan. They didn't have any interest in pursuing it. And so that kind of killed the program. And yeah. And so I left and started looking for something new.
So, what'd you do after that?
So I spent about 9 months looking for a job, 8 months. No, maybe it was 7. Yeah, it was 7. During that time, I started writing a sci-fi book. Applied for jobs and came across this consulting firm, Alacrita.
Wait. Can we go back to the sci-fi book?
Oh, yeah. Yeah. So I love sci-fi. I love sci-fi. I read. It's my favorite pastime is to just sit and read a sci-fi book. And I had some ideas for a sci-fi book, so I started writing one. And I only made it about 4 chapters in, but and then I found a job, and then I ran out of time, and it's kind of still sitting there, and I still have the ideas in my head. I still need to sit down sometime and write it, but yeah, it was about is about a race of aliens that precedes planets with a virus to basically kill off the populations that are developing, sentient populations, so that those worlds are still available to them as they continue to expand. And so, so that's what it was basically about. And I haven't quite figured out exactly how it would conclude and all that kind of stuff. But anyway, so that's what I was doing. But then I came across this interesting consulting firm, Alacrita. And from everything I read on their website, I'm like, well, this sounds like what I actually thought all consulting was like in the first place. And it's very, most of the, let's say almost everybody at the firm who's a consultant has a PhD. Because we're helping it's a consulting firm specifically geared toward helping biopharma companies develop drugs. And without a PhD, sometimes it can be near impossible to really fully understand exactly what somebody is trying to do and how to help them. And almost everybody has experience developing drugs in biopharma. And so, that experience is also used for helping clients. And so, yeah, it was a great job. I loved it. Worked from home most of the time, at least for the first year. And since 2019, pre-COVID, I haven't worked in an office. So there's really no need to work in an office. Everything consulting does is look up information online or in manuscripts or go to conferences or things like that. But almost everything can be done on a computer with a computer screen. So unless you really need to interact with people, you don't really need to go in the office. But yeah, it was a great firm, a lot of fun to work for. Very interesting projects. I ended up heading up the asset search and evaluation practice for the firm. Search and evaluation is what companies do when they've got a drug in phase 2 clinical development. They've got some very early R&D going on in a specific area, but nothing in between. And they want to bring in another asset that's, comparable to their existing therapeutic pipeline approach, disease area, things like that, but bring in something that's maybe in phase 1 or maybe 18 months from a phase 1 clinical study. And so they would ask us to help them search for and evaluate all the various assets that might be a good fit. They would let us know what their search criteria were, and we'd hunt for assets and try to find them something they could in-license. So yeah, it was a lot of fun.
So what are you working on today?
So I just recently joined Biogen, about not even 4 weeks ago. And I'm working in the external innovation group within the corporate development group. And I'll basically doing what I did at Alacrita, searching for assets that Biogen might be interested in partnering on, in licensing, things like that. Yeah, pretty cool.
So what advice would you give to someone wanting to go into the same field as you who's still in high school or very early in college?
Well, I would say, don't ever listen to anybody tell you can't do anything, because people told me, well, if you go into consulting, you'll never be able to go back to research. Well, I did that. I've also heard people say, oh, you're in research, you'll never be able to go back to consulting or do business development. Well, I did that. So never listen to people tell you you can't do anything. And the other thing is, there really is more than one road to the top. There are myriad ways to get to where you want to go. Just have a goal in mind and try to position yourself to get to where you want to go. At times in my career, I've had to pivot to something completely different, or it wasn't completely different. It was tangential, I would say. But it's enabled me to move toward the goal I wanted to go to. Like I said, virology in terms of drug development is dead. And then COVID came along. I was like, well, it's a flash in the pan. It's a flash in the pan. But it kind of has been a flash in the pan. Nobody talks about it that much anymore. It's it's pretty well controlled. Get your vaccines. I've had 7 of them. Get your vaccines, and you'll be fine. But to get to where I am and what I do requires a science background. So if you want to end up where I'm at, you do need degrees in science. It's better if you have a PhD, but you don't need one. You could do a master's in some field, science field, spend some years doing research for industry, and slowly position yourself into a BD role. That's possible. But yeah, there's myriad ways to get to where I am. You can go get an MBA. You can have a PhD and then do an MBA, or have a master's degree in biomedical science and do an MBA. And that's the same as having a PhD and going into consulting and then going into a BD role. Like there's multiple ways to do it. But having a science background helps if you want to work for the biopharma industry. So.
But yeah. All right then. Thanks for meeting with me today.
Yeah, you bet. It was great chatting with you. I hope this helps. I'm always happy to give people career advice if they want it. I reached out to people in the past and they provided it to me, so I'm happy to pay it back or forward. But yeah.
Thank you.
All right. Yeah. Thanks a lot.